Alright, so in today's video we are going to be talking about organic molecules and how we combine them through polymer synthesis to get the molecules that we see every day in our bodies. So the first thing that we need to look at before we actually get into organic compounds are the parts that make them. So we already talked about atoms and molecules and elements and all of those that combine to form the different molecules that we see around us. In the case of organic molecules, however, we have to do a process called polymer synthesis to get our larger molecules. Now, in this case, we have two parts. We have a monomer and we have a polymer. A monomer, mono means one, so these are going to be our most basic building blocks. These are going to be repeating units that we combine to form our polymers. Our polymers then the larger molecules created by joining multiple of these monomers together. So poly, we see in polymer, means many, so they're going to be formed by many different subunits, many of these monomers, to get our larger organic molecules. You can compare this to if you've ever played with Legos. You see the monomers are the individual building blocks. As we put them together, we get larger and larger polymers. Now, how do we actually get these monomers to stick together? Well, for that, we need to do a process called dehydration synthesis. Now, I like to show during lecture that this name for this process gives away what the process actually does. So in dehydration synthesis, we look at the first word, dehydration. Dehydration we usually associate with losing water. Typically, if you're dehydrated, you need to drink water, you're lacking in water. If you ever go to a hot place, usually you get really dehydrated, you got to drink a lot of water. Synthesis, to synthesize is to make something. So, minus water, and make. So we are making something by removing water. So dehydration synthesis, we are creating, in this case, the thing we are making, is a bond between two monomers. In order to do this, we need to remove a molecule of water. Now, what is water? Water is H2O. So you can see on all of these monomers, we have these OH groups on both sides. We call these hydroxyl groups. And these hydroxyl groups can be broken apart to form bonds. So you can see we have a hydrogen on this side that we're going to take off. We're going to take off the full hydroxyl on this side, take them and spit them out as water, and this oxygen then is going to be used to hold these two monomers together. If we wanted to add another monomer, again, we have this OH, another OH over here, so we'd take the OH from this, the hydrogen from this side, and we could bond it together through that other oxygen. So we can keep repeating this. Now, you won't always see hydroxyls. In the case of amino acids, we're going to find that there's amino groups that have nitrogen, but still the process is the same. We take water out to form a bond between two monomers. Now, on the other hand, if we want to take it apart, we have to add water. So this process is called hydrolysis. Hydro means water. Lyse, L-Y-S-E, as opposed to the insects. Lyse means to break or to split. So in this case, we are breaking with water. We are breaking apart a polymer by adding water. And you can see this in the diagram below. We have our water molecule, and just like we saw with dehydration synthesis, we are going to reform the hydroxyl on this side, and then the hydroxyl is going to form on the other side from what's left of the water. Again, if we wanted to do this, we could keep repeating this, so we could break down a really large polymer into all of its monomer subunits by doing hydrolysis. So just comparing the two, you see dehydration synthesis. Dehydration means to lose water, so we are going to take these hydroxyls and remove them to form that bond between the monomers. Hydrolysis, we are adding the water back in to split it apart back into its monomer subunits. So that's going to lead us into talking about our macromolecules. So our organic molecules, we're going to call macromolecules because they're typically really big and have a lot of these monomer parts to them. So we're going to find that for each of the four that we're going to talk about, there are different parts. So you're going to see there are monomers and polymers for all of them. There are elements that we're going to find in common between all of them. We're going to find that each one has its own special use, its own special function, and we're going to look a little bit at where we find them. So the first one that we are going to talk about is carbohydrates. Carbohydrates are our simple sugars. You typically see this if you have any candy or soda or fruit. Breads, you've probably heard, are called carbs. Same thing with pasta. They are going to be our short-term energy sources. You probably have seen this if you have ever done sports. A lot of people like to carbo load before games. What that means is you eat a lot of sugar, a lot of breads, a lot of pasta before your game so that you have that short-term energy that you can burn quickly throughout the game. We also find it in some structural formations such as cellulose, but mostly we're going to think of carbohydrates as short-term energy. When we look at the structure of carbohydrates, we find that there are carbon, hydrogen, and oxygen present in these molecules, and they are always found in a 1 to 2 to 1 ratio. So what that means is if we have glucose, we have the molecular formula C6H12O6. So for every 1 carbon, there is 2 hydrogens and there's 1 oxygen. Now if we combine these and we make a polymer out of carbohydrates, you'll see that rather than do this same ratio, we don't have that perfect ratio. You see C12H22O11. If it was perfect, it would be 24 and 12. So take a moment, think, where did those hydrogens and oxygens go? Okay, so we find that the reason that we don't have this perfect ratio in our polymers is because we have to remove water. We have to do that dehydration synthesis in order to form this polymer. So we remove water, it's where the 2 hydrogen and the 1 oxygen were taken. And a trick for recognizing carbohydrates, if you ever see a molecule that ends with OSE, typically that is going to be a form of a carbohydrate. Couple examples. Our monomers are called monosaccharides. Monosaccharides means simple sugar. So this is going to be 1 simple sugar. A couple examples of those. We have glucose or blood sugar. We're going to talk about glucose a lot throughout the semester. Not just in this unit, but in cellular respiration, photosynthesis, all sorts of different processes are going to use glucose. Fructose is fruit sugar. You commonly see this in high fructose corn syrup, in your drinks or sodas or things like that. Another one that we're going to talk about, especially when we get to nucleic acids, is one called deoxyribose. We actually find this in our DNA. If we combine 2 monosaccharides, we get a disaccharide. Disaccharides means 2 sugars. A couple examples, sucrose or table sugar, and lactose. Lactose we find in dairy. You've probably heard the term lactose intolerance. Lactose intolerance relates to the inability to break lactose into its 2 monomers, I believe glucose and galactose. So what happens is rather than the enzymes in your body, the chemicals in your body breaking that down into its monomers, the bacteria gets to it and causes gas to build up. And that gas leads to discomfort and stomach issues. If we combine a whole bunch of these monomers, these monosaccharides, we get a polysaccharide. So poly means many sugars. A couple examples of those, we have starch, you commonly find in potatoes. Starch is a good energy storage unit for plants, In animals, we find it in glycogen. Glycogen is our short-term energy storage in our bodies. We usually find it in our muscles and throughout our bodies. Cellulose is kind of the oddball for this. So you see it does also have that OSE on it. Cellulose is a structural material that plants use to keep themselves rigid. This is why if you ever knock on a tree or knock on wood, it's very solid, because these cell walls of plant cells are made of this cellulose. It makes them really rigid and really strong so that they can hold up a bit better. And of course, we just got a lot of examples, but where do we find this? We find it in breads and pastas. We find it in very sugary foods. We find it in fruits, milk, also in our bodies as glucose and glucose and glycogen. We find it in plants as cellulose and starch. So we find this a lot throughout the world. But like I said, the main point of this, you can see all of these are going to be very short-term, very quick burning energy sources. Our second organic molecule is the lipid. The lipid is a long-term energy storage. This is going to be your fats, your oils, things like that. They're also used for cell membranes. We'll talk about that in Unit 4. It can be used for insulation. Think bears going to hibernate need a lot of fat stores to survive the winter. Same thing, they can be used for hormones, steroids, things like that. We're not going to get too much into the hormones and steroids, but it's another good example of a lipid. In terms of structure, while they do use the same elements as carbohydrates, CH and O, they occasionally do use phosphorus as well. We'll talk about that a little bit later. But there is no perfect ratio of how these form. You can see we have very little in terms of oxygen down here. We have these really long tails that are filled with carbon and hydrogen. So this causes there to be no perfect ratio for these. And these are going to be long-term energy because of how they break down. So in our carbohydrates, we had a ring shape. We typically see rings for carbohydrates. These are really easy to break apart because all you have to do is snap a bond between a carbon to get the energy out. All of the energy is going to be stored within those bonds. Same thing in lipids. The energy is stored in the bonds, but if you were to chop off one of those tails, you lose all of your energy. So for lipids, what we have to do is we actually have to break them one at a time. And that's going to help us get that energy back out while still allowing it to last a lot longer than our carbohydrates. So these will last a lot longer in terms of energy use, so they can be more sustained than carbohydrates. When we look at lipid structure, there really isn't a monomer. We think of a monomer as a single repeating unit. There may be a little variation, but generally they're pretty repeating. In lipids, we don't really have that. We have two parts though. We have a glycerol, which you can see in blue is the backbone of the structure, and we have the fatty tails, these being the tails. We usually have 2 to 3 tails on a lipid. We can also have a phosphate group. You can see over here, this is a phospholipid. It has this little red area with the phosphate group up on top. We'll talk about phospholipids specifically when we get to cell membrane formation. Our polymer, on the other hand, something called a triglyceride. Triglyceride, tri means 3, so we do have these 3 tails, as well as the glyceride because of the glycerol backbone. The other potential polymer we could have is this phospholipid. All one word. So phospholipids have 2 tails in that phosphate group instead of one of these 3 tails. But for the polymers of a lipid, The double bonds are going to be really important. They're going to kind of determine what type of a lipid we have. We can have two different kinds. We have saturated fats and we have unsaturated fats. And the difference between these two is based on how much hydrogen is present in the fatty acid tails. So you can see in this one we have two tails that are completely full of carbon and hydrogen. You can see there's no double bonds on either of those, meaning that they are fully saturated. On this tail, however, you notice that there's a double bond present in this tail, causing it to kink, to move towards the side. And this means that for this tail, we would consider it to be unsaturated. It doesn't have the full amount of hydrogen that it could possibly have. So when we compare these two, saturated fats are going to stack very easily. They kind of lay flat on top of one another. And what that leads to is they can hold as a solid a lot better. We see this in things like butter and animal fats. Unsaturated fats, because of that kink to the tail, they don't really sit well on top of one another. They kind of move around each other. And this is going to lead them to be more liquid and include things like your oils. So your vegetable oil, your peanut oil, your canola oil, all of your cooking oils would be examples of unsaturated fats. Okay, our next organic molecule is the protein. The protein is going to be responsible for a lot within the cell. Those jobs include things like catalyzing reactions as enzymes, being structural, moving molecules throughout the membrane, defense, parts in your muscles. They're used for muscle building and muscle repair. They're used for a lot within the cell. In terms of atoms, in terms of elements, they are going to have carbon, hydrogen, oxygen, nitrogen, and occasionally sulfur. So we consider this to be CHON. And their structure includes three groups. We have an amino group, a carboxylic acid, and this side chain. So I like to look at amino acids kind of like big plus signs, and each side of the plus has a different part to it. So this amino group is a nitrogen bonded to two hydrogen. This is where one of our bonds are going to be formed. The other bond is going to be formed through this carboxylic acid group. So you can see the OH here would bind to the hydrogen on another one, be spit out, so we could form a polymer from these amino acids. You also have this side chain. Now you can see the letter R there. There is no element that actually has the letter just R. So instead, this is a placeholder, kind of like a variable. And what this means is that for each R group, we have a different structure. Now, I will not require you to at least memorize what these R groups look like, but you should understand that each one has different structure to it. So for example, glycine, you can see, has just an H. Tryptophan has a bunch of rings. Glutamine has a bunch of branches and a bunch of nitrogens and things like that. We did find sulfur in ones like methionine or cysteine. So each of these are going to have their own special structures, their own special properties, their own special functions. And these are all called amino acids. So amino acids are the monomers of a protein, and they're going to be held together to other amino acids through the use of peptide bonds. So peptide bonds are the name we give the polymer bonds in this case. And our polymers are then going to be called dipeptides if it is two amino acids, and polypeptides if it is more than two amino acids. So these are all going to behave and form very differently, as we do have 20 different amino acids. So each one's going to behave differently, they're all going to form differently, and they're all going to have different jobs based on that formation. Where can we find proteins? We can find them in meat, fish, eggs, dairy products, seeds, nuts, anywhere that you'd expect to find protein if you look into dieting. You can find protein in all of those. You can also find it in hair, nails, and horns as keratin. And like I said, you can find it in your cells and in your muscles as different fibers and filaments. Our last organic molecule is the nucleic acid. The job of the nucleic acid is to carry genetic information and store it for the cell. Genetic information is kind of our instruction guide for how to build different things within our bodies. Structures, we have carbon, hydrogen, oxygen, nitrogen, and phosphorus. And the structure is going to be built out of 3 parts. We have a nitrogenous base, we have a sugar either in the form of ribose or deoxyribose, and we have a phosphate group. So you can see even though we have this 5 carbon sugar here, it is not technically a carbohydrate because we have these extra parts, this phosphate and this nitrogenous base. The nitrogenous bases are going to be really important in identifying what monomers of nucleic acid we have. So the monomers are all called nucleotides. So I like to think of nucleotide, nucleic acid. We have 5 types. We have adenine, or A, thymine, or T, guanine, or C, or guanine, or G, or cytosine, or C. The last one, uracil, U, we only find in RNA. So we're going to, once we get to DNA synthesis, we're going to see that there are differences between DNA and RNA. So when we make DNA and RNA, the bonding rules for them are as follows. A is always going to bond with T or U. C is always going to bond with G, the way I like to remember. A and T both have straight lines, which makes them bond together. C and G are both curved. That helps them to bond together. And then A and U are going to bond together just because T swaps out with U. The polymers, we have 2, can either be DNA or RNA. DNA is going to store genetic information. RNA is going to be the messenger. It's going to send out genetic information throughout the cell. So looking at our polymers, you can see DNA is a double strand. We have thymine, cytosine, adenine, and guanine. RNA is our messenger. It's going to be single-stranded. It's going to be transported throughout the cell. It's going to have uracil, cytosine, adenine, and guanine. You can see for all of these nitrogenous bases, we have 2 big and 2 little. Same thing, 2 big and 2 little. So 1 big is always going to pair with 1 little for each pair. So that's why we always bond A to T and C to G, because they all fit together, 1 big and 1 little. 1 pyrimidine and 1 purine. Where do we find nucleic acids? Well, nucleic acids are actually used to code for proteins. So anywhere that you can find proteins, you're likely going to find nucleic acids there coding for them. So again, your fish, your meats, your seafoods, your mushrooms, your legumes, all of those are going to have nucleic acids because in order to get those proteins, they have to be made using the code found in nucleic acids. All cells are going to have DNA and RNA as well, so we find that commonly in our cells. Same thing with viruses. Viruses have DNA and RNA that they use to infect their hosts. Even though we technically would not consider them to be living, they are still found in viruses. So that is where we are going to end for today. We are going to spend the next couple days reviewing because these organic molecules are very complex. So it's good to get the practice in just while we can before we jump into anything else, because these organic molecules will pop up multiple times throughout the semester. So it would be a good idea to study these, keep practicing them, and hopefully by the time test comes, you should be ready to tackle anything like that. So if you have any comments, questions, concerns, please email me, and I hope to see you in class.